it will feed on mucus and other things that are aggregated and parasites. So we'll clean actually the surface. So this is a win-win symbiosis. So you'll hear us using these terms. So you have a parasitic is a symbiosis where one is harmed, the other one wins. Mutualistic is where they both win, and then a commensal is when one is harmed and the other is neither harmed nor gets the benefit.
Looks like it should be around 240.
240. I'm just about lined up on that, so I'll push here and see how it goes.
So yeah, it looks we're about on the move.
I'm not swinging anymore.
So as we zoom out and travel further up this ridge, you can kind of make out some layering that the lava flows have produced.
Looks good to me.
They're dipping away to the left of the screen or down the ridge to our left. To the right of the ridge is actually some removed landslide debris. So we're kind of at the ridge line where the landslide scarp originated.
Copy that. And you just want to adjust your heading to follow him and give him some time to get out ahead of you.
Yeah, if you want to adjust to something. Yeah, something like 240 if you don't mind. Now that I'm getting a little bit high here. Lots of current. So now that we're on the top of the ridge, it's not quite as really popping. Oh no, now that I say that, I see lots of things. It's not as tall.
Yeah, we see it as the diverse assemblage continues here, which is quite spectacular. Up ahead, we're about to see another one of these polyopogon sponges. Here, which is quite spectacular. Up ahead, we're about to see another one of these polyopogon sponges, very characteristic, and that's going to be a polyopogon species B. And we see a lot of these bottle brush type corals, and those are going to be chrysogorgids, and those are going to be in the species Chrysogorgia geniculata. But yeah, we see many different, we see primnoid corals,
when you get in here,
chrysogorgid corals,
be mindful of
pink corals, primnoid corals.
How you set up your shots.
And yeah,
it's possible to look
nice
into the
diversity and abundance. There's a mushroom coral too at the center.
If you put your side to the current,
so I see at least a dozen species here, and in the display right now, it's just
you know, up current. Let the push kind of push you along and turn your heading, and you'll get a little pirouette type of look.
Yeah.
So depending on how you're set up, there's two different ways. You can also fight it to the side. It's just can be a little harder.
And also
takes more effort from the vehicle.
We were getting faults in the aft flat yesterday.
There's a pink coral to the bottom. So yeah, diversity is something that we are very interested in. Diverse assemblages are something that we, as conservation biologists, are particularly interested. Diverse communities are thought to be a lot more resilient. So if you have any kind of impacts, a diverse community is thought to be much better equipped to overcome such adverse impacts. So yeah, if you have single species in an assemblage and you have some kind of impact, you could wipe all of them out. But if you have a diverse assemblage, there might be some species that might overcome an impact, others not. So it's a better way for a community to recover when they have a greater biodiversity. And so this is a focus of a lot of conservation, is to try to protect some of the most diverse assemblages, because they're thought to be also the most resilient and hopefully the ones that are able to overcome things. And Amy just pointed out a hemicorallium, a pink coral, and we're just about to go through a personnel change here in the by our pilots. I do want to thank again all of our shore-based scientists who are joining us today. I see that the group of shore-based scientist in the chat log has increased since we first started at the bottom, and I wish to think it's because people were communicating to each other about some of the early images we saw. So as soon as we stopped on the bottom, yeah, we saw a very, very dense community, and yeah, it's quite interesting. Like, this is probably somewhat comparable to what you would see in shallow water reefs in Hawaii, this kind of densities, but we are close to 1.5 miles deep. If we have a second here, Dan, there's a request to maybe zoom on one of those mushroom corals, dead center, the red ones.
They are the red ones?
Yeah, the mushroom corals are those.
I see.
Right behind the sponge.
Sure. All right, I'll do my best.
And next to it is this cactus-looking glass sponge. That's the Walteria, and that's going to be Walteria compare with Lucardi, so near Lucardi. That's the best ID.
Co-pilot, looks like you set it on 230. Is that what you think the ridge general direction is?
Stand by, watch lead, just getting a feel for the current.
Copy that, thanks.
Just go from this side, try not to.
Yeah, so there is a nice current. You can see some of those bottle brush colonies there in the back. There's one in particular, how it's kind of being pushed towards the right by the current.
The corals in the foreground.
And these kind of dependable current environments are something that we're looking at. And so these corals and sponges grow in places where you have strong currents.
Okay, let's zoom in video. I'll try to hold it steady.
Looks like you've settled on something like 230. Does that look like uphill to you? 240?
So these are mushroom corals, and looks like, yeah, the genus Anthomastus.
You want me to come down a little bit?
Zoom in a little tighter.
What's your delta?
Copy
Yeah,
that.
you could come down a little, yep.
And yeah, Scott pointed out there's an interesting kind of line of corals there in the background, and kind of all lined up next to each other and all facing the same direction.
Got an ID.
All the yeah, thanks, Dan.
Zoom out.
All those fans are all pointing into the right direction and the same direction, and that gives you an indication of where the current is coming. So the fan-shaped colonies are typically oriented perpendicular to the predominant current flow. So by looking at the orientation of these fans, that gives you an idea of where the current predominantly comes from. So in this case, it would be kind of just going across those fans.
Ready for that, pilot?
Yeah.
And yeah, and Scott also points out that in addition to
Bridge.
yeah, that interesting assemblage, that they're all those larger colonies that you see are also on larger rocks.
Can I get a ship move, please?
So something we do see in some of these communities is occasionally you will see some colonies kind of being toppled over. So it can happen that a colony will become actually too massive for the rock it lives on, and then at that point it will just topple over. So for some of these larger species to be able to persist and live for a long time, they need to settle on something that is large and stable, because otherwise they can basically outgrow their habitat.
Repeat the bailout you have? Sorry.
And we'll see if we see examples of that later on. You do see that occasionally, that some of these corals are toppled over. And
I would think that opposite of the move we're doing right now is the best bailout. Closer to...
so yeah, Amy, on your comment, that's something that we've been off and on about the event log, having the constant stream of the environmental data.
Facing 240, opposite 240.
We haven't been able to get a permanent solution to that. Yeah, I'll try to.
Hi Daniel, this is Andrea. Is there another big potentially Paris Clipper 4 on the left?
south of that ridge peak.
Possibly, yeah.
Bail out
So,
in that direction.
pilot, if we have a second, there's a
mostly
tall
east.
fan to their left that's just on the periphery of our view.
Okay.
That very tall one there, yeah.
Best bet as we go along here.
Yeah, I'm a little stretched out on the tether.
Okay.
That's correct.
I
So-
think we'll be getting closer to that in a bit.
Okay, so if that's the same that we saw earlier, there might be an interest to collect.
You got it. Yeah, we're heading in that direction, watch lead. So
Yeah,
if there's anything in the foreground,
that
just,
looks different from what we saw earlier, but I'm not sure if we've come across this species yet today.
Copy that, Andrea. Thank you.
Is there anything in the meantime?
Take maybe a look at this yellow fan to our left.
Okay.
And if it's what I believe it is, I think it's actually a zoanthid overgrowing a coral, but we'll take a look.
Okay, see if I can get over there.
Because it looks like the very tips of those branches are actually the coral, and then you have a zoanthid underneath it. We'll take a look.
Just trying hard not to make sure I don't knock over any of these other corals.
Yeah. So to the left is a pink coral, that pink-looking individual. So that's in the family Corallidae. The coral next to it, the red one, that's a paragorgia or a bubblegum coral. And I believe this yellow one that we are about to image is also a bubblegum coral that has been overgrown by a parasitic zoanthid. And
Okay, let's do a partial zoom. I'll try to hold it steady. And co-pilot, can you take my camera?
Copy.
Yeah, this is not working.
And yeah, so that's a bubblegum
Yes,
coral.
please. Yeah. The yellow coral.
Paragorgia, and then that yellow individual, that's a zoanthic and a parasitic zoanthid, and that's going to be in the genus Bulla gummy zoanthids,
Too far?
perhaps
Yeah, that's
referring
fine.
that it's bullying the coral that it's living on and parasitizing.
Got any more video?
Just a little bit, stand by.
And so yeah, it's going to be in the species Bulla gummy zoanthus
ROV now?
emilicardium.
Did you dial in that last move? Copy bridge, thank you.
And so yeah, we have several things happening here. So the red coral, that's a bubblegum coral. And that typically to have these ophiuroids, the brittle star in the middle, that live on them. And that's what you would call a symbiotic association. And then
See that video.
you also have these zoanthids, that
Thank you,
yellow
Establishing.
animal overgrowing the coral. And in fact, it happens commonly that you see the entire coral being completely overgrown and smothered basically, and eventually killed. Depth is 2,011 meters.
Check move has begun.
Just
Okay.
to give folks a heads up, so we are going to be leaving the bottom at close to 3:30 Hawaii Standard Time. So that leaves us about 4.5 hours of bottom time remaining on this dive, and yeah, we'll try to get a good blend of getting some close-ups, but we do also want to cover some ground and try to understand how far this community extends.
You get a snap.
So bear with us as we navigate through this very dense association of or aggregation of animals. We won't be able to image every single one of
Still
them.
pretty stretched out video, but you want to do a snap zoom, and we'll make sure that this is something they want to see closer. And I'll try to hold it steady for you.
Yeah, so that is definitely, I agree with you, Andrea, that is different than what we saw earlier.
It's actually missing a couple branches. Oh, off the other side.
I think that looks more like a Norella.
That's full.
Okay, so this is something we're interested in.
Yeah, I don't think so. Now this is something different.
Okay,
Yeah.
zoom out.
Yeah, we can go ahead and continue with the move.
Okay.
So yeah, if you joined us later in a dive, we're diving at a depth of 2,000 meters on a seamount that rises from 4,500 meters to about 1,000 meters of the surface. We are 1,300 miles away from Honolulu, about 70 miles south of the boundaries of the Papahanaumokuakea Marine National Monument. And this seamount in particular is interesting because it's very isolated. The closest, a lot of times you have seamounts all occurring in dense clusters together.
Sure.
But this is different. This seamount, the closest one is one seamount we dove on yesterday, which was close to 60 miles away. And then the other close seamount is another seamount that's to our south, that's over 70 miles away. And and that's the seamount that we're going to be diving on tomorrow. So yeah, we're looking at some of these seamounts that are widely spaced apart.
Okay, looks like we're feeling that move now. So.
Yeah, and the contour should sort of curl up north as you're going along. We'll chase you up that way.
All right.
So yeah, this is absolutely wonderful to see. This dense community. This is full of life, and that's always something that mesmerizes people when they see these images of the deep sea for the first time, is how much life there is. And that massive sponge right there, that's a glass sponge, and that's going to be in the genus Poliopogon, and that's going to be this Poliopogon species B. So that genus of glass sponges, right now we have these place-based, these kind of placeholder names.
Careful of the tether hitting that rock.
And they're distinguished based on how many of these atrial surfaces they have. So they have these vertical surfaces on top of their tissues, and how many of those they have is what we use to differentiate them. But yeah, this is spectacular.
Which face?
The one I'm looking at now, because he's facing 240, so that's just to the left of this big rock feature. And
So I'll have to.
So yeah, we see a little local high in front of us, kind of like a small pinnacle, and a lot of times what you see is on pinnacles like that, there's a lot of life. And I think there's a little sea star to our right bottom. There might be some interest, if that's what it is. On the coral there, and I think there's someone is taking a little lunch, which is appropriate. That's about what's going to happen here on the ship with a lot of us. So this is, there's a couple of genera of sea stars that are common predators.
Okay, let's zoom in video.
Hey Nav, can you center up the ship there in IPAC?
Of course.
Thanks.
So yeah, you see a bamboo coral that's being preyed upon by the sea star, and that's something you see somewhat commonly. And you actually, if you were to zoom in from the other side, you would see actually the sea star averting its gut onto the surface. And
Can you take my camera again, co-pilot?
Copy.
Just tilt up.
And I believe this is going to be in the genus Hipposteria, and I believe this is the genus, the species Hipposteria muscipula. I don't know if there he is.
Hello, it's Chris Ma.
Excellent. I was thinking of you, and I was about to call you out.
This looks like a very handsome example of Hipposteria muscipula, which is that species that we were seeing at deep depth last year in this area. Was the one that I described last year. Was a new species, and it's feeding on a bamboo coral, right?
Yep.
Yeah. So that seems about right. This is also that same one that we noticed, that the red one that had the two little scale worms living on it. But it's looking more and more, I guess, since yesterday that those worms might not be necessarily specific, but who knows? But yeah, the characteristic surface has all of the really well-developed spination, and the arm and body shape looks about right. So. But Hipposteria is a genus that you see even in shallow water of other species. You know, in colder areas, colder waters, for example, in the North Atlantic and in the North Pacific. There's one widely known species, Hipposteria phrygiana, and then there's this weird deep sea one, which occurs not only now in Hawaii but New Caledonia and New Zealand. So it's quite wide. A lot of these things are quite widespread. So it's really interesting to see how they occur because there doesn't seem to be any pattern. You know, there's Calliaster, and then there's Hipposteria, and then there's Callia, and then there's Evoplasoma, and you know, I'm not quite sure how they settle out. You know, why there seem to be more of them in some areas than others, because it's not necessarily the food, or maybe it is, and it's just, you know, we're not seeing enough of the data. But anyway, thanks for that really brilliant footage.
Yeah, and thanks for
watching. Thank you.
Thanks for calling in. So yeah, that was Dr. Christopher Ma joining us from the National Museum of Natural History, Smithsonian Institution in Washington, D.C.
35 meters bearing 270.
One of the most
please.
Taxonomic authorities, world's experts on sea stars, and as well as other groups of echinoderms. So yeah, thank you very much for chiming in.
Good copy, Rich.
Sorry, I can't quite hear you, Tara.
I'm just going to hold this for a moment and then zoom a bit closer.
Yeah, hi Daniel, this is Andrea. I'm just looking at the chat room. I wonder if this is a potential candidate for collection.
Yeah, so thanks.
There's a few others on the right as well.
So yeah, the one we're looking at right now in display, Andrea, or the one behind it? From here, it looks like they might be the same, but
yeah, I think this is the one that's of interest to Amy.
So yeah, I'll put it up here. If there's any other opinions, so Amy had expressed interest in collecting this. I don't know if Chris or Scott or anyone else or Tina want to chime in.
So they're interested in collecting this pilot, possibly?
Yeah, Chris stepped out of the ECC right now, but I think earlier we were discussing if this was the same potential for it, it'd be a good candidate for collection.
All right, so I think we have a consensus, and yeah, we're going to go ahead and collect that.
Okay, let's zoom out video.
Thanks, Andrea, for chiming in, and thanks Amy also for pointing it out. So we'll get this sample. So
they could either if you use the
no worries, thank you.
You want to make sure you have a tow hold, right?
You don't have to.
You can put in autos and
see how autos.
It's not like there's going to be a lot of mess. It's not a rock, so
right. What I would suggest is getting to where you think you might be collecting if you're in autos, and see if you have bottom lock. That's the first key
thing
to check. If you don't have bottom lock, you won't be able to, but it looks like you will.
It's a pretty hard surface below us. So.
Yep, hard surface, and it's not too far away.
Are we full wide video?
Yep, we are.
Okay.
So yeah, we're just about to set up. We're going to try to collect a sample of this primnoid coral.
Should be still catching up to you a little here. So
We'll be able to put a toe down on the rock facing into the current. That might be the better way, huh?
Well, certainly the other option, whichever one you can line up. You get a little tether here, and you can try getting a toe hold and see how that goes. Either one is totally up to your preference. What you feel is
so yeah, this is a very exciting moment here. So there's a lot of excitement by several of our shore-based scientists who had pointed out this very large fan. This is a primnoid coral, and they'll be seeing several of them. And it's also a good collection because once we don't know exactly what species it is, we just think we know what genus it is, and then it also appears to be one of the more common species here. So that's a good way of characterizing the community. You see several there in the background that appear to be the same species, and so today's dive is on a little-known seamount, Castellano seamount. So it does have a name, but up until yesterday, we only had half of the surface of the seamount mapped. And so our mapping team, a very hard-working team, spent the whole night trying to cover, and we now have almost full cover of the seamount. So yeah, the team of the Okeanos Explorer yesterday mapped a whole portion of it, and in the process came across this ridge. So we didn't know this particular ridge existed before. And so in the early hours of the morning, we actually changed the dive plan. And we're all very delighted that we did so, because this is absolutely a spectacular dive site. An unbelievable density of corals and a nice diversity of corals, and also many corals that we don't know what they are. So a lot left to be discovered. And yeah, Amy Bacco-Taylor from Florida State University, who's particularly interested in this, and had pointed it out. So yeah, as mentioned, we have a tentative ID for only the genus, so we don't know what species that is. So it's quite likely that this is a new species. So even if it is that genus, it would be a significant range extension for this particular genus. So that genus was only known to depths about 500 meters shallower, so that really gives us a nice extension. And that's also something that we're trying to accomplish with this project is in Hawaii and the waters around Hawaii. We've been fortunate to have the Hawaii Underwater Research Laboratory operational since the mid-'80s. Which had access to a couple of manned submersibles and ROVs, and Hurl has been absolutely phenomenal in documenting some of the deep water resources for the region. Now, their submersibles were limited to depths of 2,000 meters, so up until these expeditions, we had virtually no data about what's found at depths below that, and this is where the Okeanos Explorer comes in. Our remotely operated vehicle and the vehicles that you're seeing the video footage of, they are able to dive to depths of 6,000 meters. And so we're able to, for the very first time, take a look at what's below these depths that we have surveyed in the past. So yeah, on this particular expedition, we did our very first dive of it. It seems like a long time ago now, but it was just over a week ago. We dove off Mokumanamana, and we dove to 4,000 plus meters, and that was only the second deepest dive ever to be conducted in the northwestern Hawaiian Islands. And came across several very unusual animals, including a very peculiar octopus that's really captivated the attention by a lot of scientists. It's at the very least a new species. People are speculating that it might be a new genus, and it's also captivated a lot of our audiences and the general public. So yeah, as we're about to set up here, our current depth is 2,007 meters.
Yeah, just get your joy lock in there, and then you can tune your auto heading still, even if joy locks in.
That's true. Okay, because it's hard to.
Water temperature
Not
is a
twist the
chilling
heading.
So now you
2
can just
degrees
rotate
Celsius,
heading to fight it.
so just
now
2
that
degrees
you're
above
stuck to the wall.
freezing. So very
That's better, yeah.
cold water temperatures. And that's
You
something
can
that
see
we're always
a little
reminded. So when
differential,
these
so it's
samples come back up
so
to
you
the
just
surface,
keep an
and
eye on
we
that.
the collection
it all
buckets
visually
actually
from
come back up full of
But
water,
you're
and
slowing off no matter what.
every time we come to retrieve the specimens,
I would
you're
try
reminded
quickly
of
with
how
auto
cold
XY.
the deep
Yeah,
ocean
Auto
is.
XY. Okay.
This is such a slippery surface, and we don't want you to crash into the coral or the wall.
Yep. Okay.
So yeah, for folks who have been following this expedition, today is our 7th
So
dive
get to a
of
height
the
you think is
expedition.
good, Put in auto depth.
We're diving about 70 miles south of the Papa Hanaumukua Marine National Monument. We're just outside the boundaries. And we have our final dive of the expedition will happen tomorrow, and that will happen in a seamount that's about 60 miles south of us. And after that dive, unfortunately, we have to depart the area as we
So you
have to
think
make
maybe
it
just
to
not cropping against the
Kwajalein
rock?
Atoll by next Friday, and we have to cover close to 1,500 miles of distance in the process. So just going to navigate across a very large stretch of ocean. And so yeah, this cruise will end
Try to get your face in
next
there
Friday.
a little bit at least.
And but that's really only the first expedition of this field season. The Okeanos Explorer is going to spend 6 months this year in the U.S. Pacific. After this expedition is going to spend some time in Wake Atoll, mapping the area. That's where it's completely unmapped and uncharted territories. So there'll be a mapping-only cruise around Wake Atoll, which is part of the Pacific Remote Islands Marine National Monument. After that expedition, the Okeanos Explorer is going to sail west and head towards the Marianas Trench, part of the Marianas Trench Marine National Monument, exploring some of the deepest parts of the world's oceans. And then the Okeanos Explorer is going to come back throughout the Pacific Remote Islands and ending the field season in September in Hawaii.
Who knows how old this coral is?
Hey Nav, just a heads up, there's as you know, the current's really strong, but it's keeping me from keeping heading real well.
Roger that.
How's that? Is that a good distance, or am I too far away still?
Look at your sonar. I would lock it in.
I do want to take a
Light it
time
up
to
like
welcome
you
some
like
of
it close.
some of the viewers who might be watching the spectacular
We'll lose
footage
0.5%
to many of their public viewing displays that have been set up around the country.
Kick in.
In Hawaii, we have 3 major ones. We have partnered with the Moku Papapa Discovery Center. There's a wonderful educational center we have on the island of Hawaii. Also with the Maui Ocean Center, one of the most beautiful and spectacular aquariums we have in the state of Hawaii. I urge all of you who have not visited the center to go. And then also the Waikiki Aquarium on the island of Oahu, one of the oldest aquariums actually in the country. And even though it's a small facility, really one of the older ones, and we have a lot of knowledge there. And so, yeah, welcome to all the viewers who might be tuning in from there. You're looking at live video
This is too
footage brought to you about 1,300 miles away from Honolulu.
I think I'd be able to.
And we're diving over a mile deep. We're
I'd be
about
able to grab
2,000 meters on a little-known seamount by the name of Castellano.
Position step way down from 3 meters. If you can use the keyboard if that's too small. And actually, we're coming now already.
2.5.
You're gonna want
And then I also want to welcome all of our classes that are following this. We've received several emails by different elementary, middle, and high schoolers who are joining in on the excitement and have been tuning in. Very glad that you are as excited as we are about ocean exploration.
autos.
if I just adjust.
Why don't you swing around and look at it from this side? See
Okay.
if that's any better on a tether tug.
Okay.
What?
So watch, Lee, there's a lot of current here, so we're trying to set up to safely grab this sample
Copy that.
for both the vehicle and the coral.
Serious, move farther.
Yeah, copy that. So
Which
yeah,
way do you have D2 facing? Or which way
pilots
is
are
D2
just
to center up on it?
trying to
Three
set up
something?
to collect this sample of
300,
permanoid
I could
coral.
move you a
And,
little closer if you'd like.
there's a
Okay.
quite
Yeah,
substantial current here, and
303
so
as well. Trying to, but it
we're
keeps
trying to set
Okay,
up to
yeah,
collect
I'll-
this sample.
Yeah, I'll bring you in a little bit.
Copy.
So I think this might be better, because then at least if I lose position, I'll get blown away from it.
Your back is into the current, so you get blown into it.
And yeah, that's a nice illustration. You can see how
Bridge,
those
ROV nav.
those branches are dancing in the current, and here it gives you an
Can we get a
indication
ship
by
move,
the
please?
pretty substantial
Let's do
current
20
flow.
meters, bearing 310 at 3/10 of a knot. Thank you.
So there's a question here in the chat, Luke, about the tiny objects on the
Affirmative, thank you.
rock, and those were tiny barnacles. And yeah, we at one point we
Okay, we're
zoomed
trying
in,
to
and
bring Sirius
so
in a
those
little closer
are
for your collection here.
barnacles. So
And
barnacles are a group of organisms. They're particularly interesting, so they're somewhat closely related to shrimps, crabs,
I think that
and
Sirius
same
just
group
ended up
there.
settling a little farther than I thought.
And that
so we're
was
gonna actually
unknown
bring
for a
them
very long
closer here.
time. As when you look at them, actually, they look more like a maybe like a mollusk, because they have those
where there might be
plates.
Interesting.
And so for a long
Okay.
time, scientists actually
Take a
believed
look
that
at it, keep the
barnacles
video
were
interesting, and then we'll come back and take the
Okay.
sample.
All right.
mollusks, and it was not
All right,
until
can we brighten it up,
scientists
video?
first started
Sirius, a
seeing
little time to-
We're going to bring Sirius in a
the
little
larvae
closer.
of these organisms that you
Oh,
see
yeah.
Can
the
you
resemblance.
brighten it up? It's pretty
Yes,
dark.
of course.
They have these segmented
Thank you. Maybe I can get my toe down
sections.
there
So
and
they're
look
the larvae.
Be
So
interesting
very
to
very
check out
similar
like that.
to
certainly try it.
crabs, shrimps.
I'm really concerned that if I lose the autos,
Another
or if
interesting.
my toe slips, then it's just going to plow into the coral.
So barnacles,
Okay, wanna try that?
Ceropedia is the
Yeah.
scientific name for that group, which means curled feet. And a lot of times when you zoom in, you actually see those curled feet coming out of those plates, and they will use those feet to basically expose that into the water and feed, capture tiny particles that might be coming their way.
Try to come back.
So
Okay.
they
Get
are
something around below,
kind of upside
settling on
down,
this one,
so
get
they're
a little
attached
soft.
to the substrate by their
Back.
forehead and using their feet to feed.
Right now. Watch lead nav.
Go ahead, Nav.
We're just trying to get Sirius. There's quite a bit of current. Sirius is acting a little different than we expect. We're just trying to get Sirius in a little closer so we don't feel the tether bob for this grab. Pilot's going to back up and look for something interesting here in the foreground until we get that to keep the video interesting. Is there anything?
Copy that.
Got this big sponge down here.
Maybe the smaller one, the
Yeah.
big one. I know what it is. I think. So yeah, that large sponge. That's a glass sponge in the genus Poliopogon. And the other sponge is also a glass sponge, but as we zoom in, it might be in a different genus. We'll take a look here.
All right, let's go in video, partial. It's actually going to be the small one up to the left, and I'll try to bring it around.
And now that is the same. So those are both Poliopogon.
We're still feeling the
Yeah.
tether bob. We're bringing it in for you.
Okay.
This should all get better.
Yeah.
Ship's almost done, so hopefully Sirius settles in. And if not,
And then,
pushing.
yeah, if you still need time, there's a coral like right to the bottom, kind of irregular bushed.
Which one?
Not the right. So there's three in view. The one to the furthest left.
Okay. That we can do. Yeah, I'm feeling it again. There we go.
So if you sidle port and turn your heading right, you'll get a little tether.
So now, there's a question here. So there's a pretty strong current, and we're just getting the vehicles lined up to get the collection done. In the time, we'll have a little bit of time until Sirius catches up to us, and so we're just imaging a few of the animals in the vicinity until we can be properly lined up to collect that Paracalyptrophora.
Maybe we could drop down a little, co-pilot, and give him some slack.
Going further.
So the coral to the bottom, that's a Chrysogorgid, and the one that we are...
Datsu is telling me negative, because it looks like the tether is going above me.
Oh, okay. I see what you're saying. Okay.
Okay. Zoom out.
so that looks like a Chrysogorgia stellata.
Okay. I think once we get the
And
ship a little closer,
in the
that'll
background,
hopefully
I
pull
was
off to the
also
side.
able to see a crinoid
Out
that
of our
was
way.
on the rock, and that was going to be in the family Antendonidae. They have these very widely spaced arms
I've got a
that
little more
are
flexibility now.
characteristic for that family of crinoids. And
So
crinoids
Sirius, is it swinging?
are
in a
another
different
one of
direction?
these suspension.
Sirius is doing its own thing. Separate from the ship.
So we're just so there's a couple of
Sirius
questions in the
has
chat. So we are just lining up for the collection, Amy. So
I can
it's
give
going
you
to just
another
take a
bump.
while till Sirius catches up.
We've done 20 meters with the ship, and Sirius hopefully is going to catch up. But Sirius is still swinging across, just coming right across the ridge. It's not really moving closer to you.
It's basically just swung around you.
So
Interesting. Okay.
we can try to.
And its heading is everywhere.
Yeah. Well, that's because you're yanking on him. That pulls your heading off pretty hard.
So we're pulling now. It's still too far.
All right. We'll
So yeah, we're
come in a
going
little
to
closer.
on these dives. We have been limiting our collections to 2 samples. Part of that is because of the storage space of the vehicle. And part of it is also because there's a trade-off on these dives.
Bridge,
We
ROV
do
nav.
want to collect samples because we realize it's very important to ID the individuals, but we also want to cover ground and be able to say something about the community as a whole.
Right.
And so, yeah, there's kind of this trade-off, and we're trying
Where
to
do you have D2 now? Farther even north?
attain multiple objectives.
330?
Characterizing the assemblage overall is the main objective,
Jeff,
and so that is of course done by close video ups,
You're gonna get
by
one good
covering
heat, you're
ground
gonna
so
get
you
a
know
yank.
how far these communities extend,
But
and then
this is
collecting samples because
certainly in range.
you need the sample at hand in most cases to be able to
Propeller's
identify
not gonna
the
pull lower down.
species even
You're
to
gonna
a
get-
coarse level.
See his heading getting turned off. We're still getting yanked on. I'm gonna put in a ship move closer to you.
And yeah, beautiful
try to
view
get
here of this
Sirius lined up on
Paracalyptrophora.
Okay.
It's just
I agree.
I don't want you reaching out and then get a yank and then.
I'm gonna lose my footing, and
Yeah.
I'm gonna be ready to pull off in case we see a heave.
Just make sure you lock in your auto heading. Bridge, ROV nav. Another ship move, please. Let's do 20 meters, bearing 330 at 3/10 of a knot.
And so just to give our shore-based scientists as well as our other viewers a heads up, so today's dive
Good copy, Bridge.
is gonna continue until 3:30 Hawaii time, so we'll remain on the bottom for another 4
Sorry,
hours.
pilot, this is taking so much
Uh,
to catch you.
we
It's a
have
good learning experience
we're
for
about
everybody.
2 hours into this dive, and so
Sirius-
after we're done with
Sirius
this collection, we'll
here.
try to really
We did a
cover up the
310.
slope.
Instead of moving there,
We haven't really
Sirius went this way.
climbed up that much, and we do want to get a sense for this community and how far it extends.
So it shifted.
So, yeah,
just
one of
swung
the-
around the ridge, huh?
Yeah,
That's
yeah. Sirius is doing something rather strange at the moment.
really strong current, I guess.
He's whipping. So you'll have to be careful of that as you climb up the ridge. I might feel it.
And I'm losing it.
There's the yank. on those holds you can get mostly vertical down and not so much into the wall, it'll be safer if it lets go. So, you know.
Can you repeat that?
When you're towing into the wall for samples, if you do mostly vertical down and auto heading and not so much
joy.
Not so much, well, you know, a joy lock still, but joy lock down and an auto heading rather than so much into the wall.
Okay.
Because if auto heading lets go with you into the wall, it can
Okay.
do strange things. Dangerous things. Sorry. So,
Some more down on the verts.
you know, I don't mean to say apply more vertical down than you already were, just do less into the wall if it's possible.
Well, actually, you've provided us with a great learning experience.
Okay. Hey, Dan, let's just back off until we've got tether.
Okay. All right.
I know you really want to settle up and grab this, but let's really just get away and wait till we figure out what Sirius is doing.
Okay.
Every time you go to settle up, you're just that much more likely to do something funny.
Okay.
Being stretched out at the end of the tether is just not good for. Part of the reason Sirius may be acting the way it is if we've been yanking it this whole time against the current, and then each time we move, we give it a little tether, and it can just swing around. So if we just give it its tether and see where it goes, then we can figure it out. But Dive Super just said that this might just be too high current of a spot to be.
Trying to.
So
Okay.
we're gonna sort this out for you here, and hopefully Sirius will cooperate and give you the slack you need to at least get this grab.
Okay.
So
Understood.
yeah.
Okay, let's zoom in video on this coral.
Sure thing. Okay, stand by.
Oh, I lost it.
Yeah, he's Delta 18 at this point. Well, I dropped a target. Why don't you sidle port, get over there, get
Okay.
yourself some tether.
Okay.
We'll just have to come back here, and once we sort out Sirius, it's just give yourself the tether. You know, the more you yank and pull, the more you're gonna get frustrated, and the more difficult it is for us to sort out what Sirius is doing. So let's just sidle over, get lined up. Try to find something interesting while we're at it.
zoom in video. Okay, zoom out.
Well, my heading isn't going quite as crazy as it was.
Yeah, so I had the pilot back towards you, give you your slack. What I think was happening was pilot was fighting really hard to keep that coral in view and pulling you against the current. And every time you got to move, you got pushed by the current rather than moving towards DT. You were just getting more slack.
Hey, Daniel, this is Andrew. Did we lose you from the telecom?
Now that we've backed off.
Hey, Andrea. So Daniel stepped out to take a quick bite to eat for lunch while we set up to make this challenging collection. Is there anything I can do for you?
Where it is. And now...
We just didn't hear you anymore, so we weren't sure if we lost you. So this, we're still setting up for collection. Is it a difficult collection to make? Should we move to another one? I think there was a few more that were on a rock towards the right.
Depending on where you are with respect to the ridge.
It's a challenging
I guess
collection.
we just don't know what's going on.
Yeah, so it's a challenging collection for the pilots due to the higher velocities of currents around this feature. So we're trying to set up and make that collection, but
So just be careful not to yank on his tether, on the tether at all.
definitely
Keep the tether as high as you
a
can.
learning experience for everybody. So still
we're
a
gonna
still a valuable collection to be made, I think.
and it looks like one final due north move hopefully will get us there.
Okay.
So if you see on top of the rock as well, I mean, all down the kind of towards the left, there are several of these colonies. I mean, now it looks like you're in good position. We can't hear the pilots in this room, so if they think it's doable, I guess, and we're ready to go, then we might as well go for it. Since they've taken all this time and energy to set up.
Bridge, ROV nav. One last ship move here. Well, not the last one, but another ship move. 25 meters, bearing 360 degrees at 0.3 of a knot.
So, Andrea, sorry, I was talking to our dive super for a little bit. He was explaining why this collection is very difficult, so I missed your last comment, but I did hear you over the headset. Can you repeat what you were just saying, please?
Lat, and it's
I was just saying that there were several more along the rock. You know, you kind of can see them all the way in the background. And if this was too difficult to do, and yeah, we could try for another one. I also was just saying I can't hear the pilots, so we didn't hear the discussion on what was occurring.
Okay, yeah, so I was just discussing with Carl, the dive supervisor, why this collection is so challenging, and I don't think any others in the local vicinity are going to change that, just because what's going on, it's apparently only happened once before. There's differential currents between Sirius and D2 and the surface where the ship is, so we're trying to balance this delicate dance between the ship and the two bodies at the seafloor. So the ship is actually being pushed north, or we're trying to move the ship north while it's being pushed away from Sirius that's moving west, and so we're trying to get the tether close enough to Sirius where we can set up and make this collection without being more or less pulled away from it while making the collection. So that's why it's becoming a challenging orientation to set up, but I don't think that that's going to change due to, like, if we move, say, 10 meters up. So So yeah,
I see. Thank you for that.
yeah, no problem. It's very interesting to watch, and it's very challenging for the pilots, but they are very talented and doing their best to get us set up. So
Yeah, they're absolutely talented.
Now, so I think I want to bring this up. So yeah, considering that we've seen several of these, I do think maybe we should move on because we'll probably see this again unless you think it's I don't want to spend too much time here setting up.
Okay. We just dialed in. I don't know if you've been.
Yeah, just
I would agree with that.
Figured out what. I don't know if you figured out what's been going on, but.
Try to get you 45 degrees under. We'll just keep going until we get there.
So what was happening is we had a big shift in current as we came over the ridge up in the water column. So as we were moving, Sirius is actually moving 90 degrees from our ship track. So this is not necessarily us setting up for the coral. This is us figuring out the currents and getting Sirius lined up with D2. It's something we would have had to do to go up this ridge anyways. So once we sort this all out, we'll be able to move up the ridge. He'll grab the sample and we'll run up the ridge for you. Is that all right? Okay, good.
Yeah, it did swing on the other side of the ship, didn't it? Oh.
Yeah, and it still is, actually, now that I'm looking. It was right in the fantail when I last was, after the last move. It settled right on the fantail. Now it's about as far as we just moved off the fantail.
So Andrea, you had a question. So the pilots are just setting up. The currents have shifted, and so it's several maneuvers that are actually required to get us up top of this feature, regardless if we collect a sample or not. So they're just lining up. So we'll take some time to
just make sure
get everything lined up.
Your delta's 20 and you're way out there. You don't want you tugging on them.
Okay. I'm not bringing up anymore.
Oh, sorry, I'm talking to pilot.
Oh.
Talking to pilot, yeah, sorry.
Yeah, he's pretty far up, yeah. Okay.
So just make sure that you just be mindful of the tether, and you don't want to be yanking it up into Sirius's face or anything like that.
Sure.
Let's see if that starts to settle in at all for you. You see side of the collections. Off to the right.
Yep. It's just on the side of that face there.
Yeah. Any of those might be the same thing. You might.
So while we're taking some time to get set up and oriented the right way in order to continue our survey up this ridge, I thought I might take the time to explain a pretty interesting conversation I've been having with Brian Boston at the University of Hawaii to explain some of the regional geology in this area. So to our right and kind of into the darkness of the abyss that we're looking at right now is a very steep cliff, and the steep cliff is basically the head wall of a submarine landslide, or basically the oversteepened parts of the cliff that resulted from that landslide detaching from this ridgeline and being moved away from this point that we're at. And so what we've been noticing along the dive while all these beautiful biological communities have been incredibly dense is we've seen some obvious lava flow layering that is dipping to our left and out of the screen right now, as well as a lot of fractures that are running parallel to the ridge line. And what we think that these fractures are indicating is that there is a propagation of fractures that is going to our left, and these propagations of fractures result in these larger blocks to become mobile over time and slide down the other side of the ridge due to the weakening of the integrity of the structure along the ridge of the landslide. So some very interesting geology as well as biology here. It's just taken a little while to kind of piece it together, and really appreciate Brian Boston discussing that with me a little bit and helping me kind of put the regional geology together. So very, very neat story, and these submarine landslides always generate some really